Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Oncology Clinical Development Optimizing Value Through Implementation of a Patient-Centric Holistic Endpoint Strategy. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 26,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 60% of all FDA drug approvals since 2012. Now, it's my pleasure to welcome our speakers for today's event. And first, I'd like to welcome our co-moderator for today's event, Dr. Todd Rudeau. There he is. He is the Executive Vice President and Chief Medical Officer at Clario, providing medical and scientific leadership across the organization. He has over 20 years of clinical cardiology and pharmaceutical research experience with a career predominantly focused on drug safety. His team provides expert consulting to clients on scientific and regulatory strategy and ensures Clario's product portfolio is scientifically robust, generating high-quality clinical trial endpoint data. And next, I'd like to welcome Dr. Anu Banzal, Vice President of Medical Imaging Oncology at Clario. There he is. As Vice President of Imaging in Oncology, Dr. Banzal provides leadership and oversight of all aspects of imaging in Phase 1 through Phase 4 drug and device trials. From protocol design through registrational submissions and post-approval monitoring, he uses his 20-plus years of experience in clinical research and patient care to support Clario's cross-functional teams and clients in medical, regulatory, and scientific affairs. And next, I'd like to welcome Dr. Thomas Mull. There he is. He's a senior scientific advisor at Clario. Dr. Mull has been working in the life sciences industry for more than 20 years and has held various positions both in the CRO space and within big pharma. Over the years, one of his areas of focus has been post-marketing projects that require patient experience data for regulatory decision-making. And finally, it's my pleasure to welcome Dr. Fabian Chen. He should be coming on right there. He is a Deputy Chief Medical Officer, Cardiology, and Vice President at Clario. In addition to providing leadership for Clario's Cardiac Safety Department, Dr. Chen provides expert consultation to clients on scientific and regulatory strategy. He has nearly 20 years of clinical cardiology and pharmaceutical research experience. Now, with the introductions out of the way, it's now my pleasure to pass the mic over to Todd. So, Todd, when you're ready, you may begin.
Great. All right. Well, thank you, Sonia, for the very kind introductions. Thank you to all of you, our quite sizable audience today that's joined us as well. I'm excited about this one as there has been so much evolution in the past decade with endpoint solutions and strategies in oncology development. And so we'll take you through this agenda today, covering the evolving role for centralized medical imaging in oncology studies, as well as some of the innovative methodologies that have more recently become accepted. The value of patient-reported outcomes and what they bring to an oncology development program, and the different technologies that can be used to achieve this without introducing excess site and patient burden. And then we'll go through some of the unique challenges to assessing cardiac safety in oncology, how to appropriately tailor a risk management strategy, for your development program. You can go to the next slide. First, just to get us kind of an understanding of this audience's perspective, we asked you all by survey on LinkedIn over the past week, what do you think about today's oncology study endpoints? Are they evolving fast enough to meet the changing needs of regulators, of payers, and patients? So you see the results here on the screen. Generally, this audience was fairly positive. The majority feeling that strategies we are implementing today are hitting the mark. And that's great to see, actually. Some were less bullish and have concerns that we may not be keeping pace. And as mentioned in this webinar, we're going to walk you through today's endpoint solutions and strategies, and then regroup at the end for some Q&A time. So do enter your questions, please, as we go, either in the chat function or in the LinkedIn Live tool, if that's where you're viewing this today, and we'll do our best to get to everything at the end. So with that introduction on the next slide, I'm just going to take a couple of minutes to set the stage, kind of highlighting some of the challenges we all face in oncology development. I'm doing this as I think it's important context to understand the why and the how as to the solutions and strategies that have evolved over time. So in other words, what's difficult in this space and how are we addressing it? Well, one of the biggest challenges to interpreting oncology data is a lack of healthy volunteer phase and lack of placebo-controlled studies. Understanding causality for adverse events, teasing out the role for concurrent treatments or historic treatments, and the contribution of pre-existing comorbid conditions, all significant challenges. Additionally, these studies can last many years, often with open label extensions for patients that are responding well to therapy. And that introduces particularly unique operational and budgetary considerations as well. And the regulatory approval strategies are quite different, with pivotal submission data often coming from phase 2, accelerated approval strategies that will require ongoing confirmatory work post-approval, all unique elements here. The patient population is also quite unique. Generally, exclusion criteria in a study protocol within other therapy areas allows sponsors to enroll relatively clean populations free of major comorbidities into their studies. But oncology is different. Very sick patients with multiple rounds of prior treatments and all the complications that come from those past treatments, they do come into these studies. And so the adverse event rates are much higher because of this. But additionally, for these sicker populations, we want to be even more cognizant of patient burden and applicable for any of the technologies that we may want to deploy within a study. So there's a balance to be struck, in other words, between increasing the scientific value of a study by adding additional endpoints and visits versus patient centricity and even recruitment and retention feasibility as well. Themes you'll hear about today. And lastly are the unique nature of the medications we study in oncology. So cytotoxic and small molecule treatments, very much still relevant, but other mechanisms including biologics, immunomodulators, targeted therapies to very specific genetic mutations, radioligand therapies, hormonal modulators, all of these make for each their own challenges and a very interesting space to work, actually. So on the next slide, I'm going to hand over here to Dr. Banzal, who leads our oncology medical imaging group, and will take us through the major considerations for centralized core image acquisition and interpretation in cancer studies, and also will highlight some of the newer innovative elements as well. Anu?
Great, thanks. It's a pleasure to be able to have the opportunity to speak to everyone today and go through medical imaging in oncology drug development. It's a broad topic, and so I have a little bit of an agenda here for what I'm going to try to talk about in the time we have. First, just go over the value proposition, why central imaging makes sense, what it's important for, and then talk about differences in implementation by phase of study, cover a few of the more common oncologic criteria, And then I want to pivot a little bit and just talk about a couple topics that we get a lot of questions on at Clario, just from different sponsors and other folks. And one of these is around site feedback. The questions around this have increased a lot since the new ICH draft guidance came out last year. I want to talk a little bit about the use of clinical data and how that can help improve blinded independent central review process overall, and then just close up with some trends we're seeing in imaging. So if we can go to the next slide. This is a quote from the FDA trial guidance, but basically all the regulatory authorities have come out with pretty definitive statements saying that in oncology drug development, a blinded independent central review should be performed for response assessment. So it's not a mandate, but FDA, EMEA, and other authorities have said for specific reasons, which include less bias compared to an investigator who may know about the patient's current clinical status, who may have additional information, they may know what treatment arm the patient's on. So putting all the reviews into a central place helps remove all that type of bias. The other thing is the consistency is really appealing to regulatory authorities. When you have 300 sites for a global phase 3 trial and you have different people reviewing the patient's imaging at each site, and sometimes even at each time point it might be a different patient, versus when you have a central review, you have some number of radiologists, 6, 8, 10, depending on the size of the study, that are reviewing all the imaging for every patient. It's the same patient has the same radiologist all the way through the trial. That consistency is really important to regulatory authorities. So that's the reason to go down this road in the first place. Can we go to the next slide? So here I want to try to hit on just some of the differences we see in the central imaging review process by phase. Endpoint definitions are on the side. They're probably pretty familiar to most people: objective response rate, progression-free survival, and so forth. In terms of phase 1, the primary endpoints, though, tend to revolve around safety, pharmacokinetics. If it's a radioligand therapy, then dosimetry, which, again, I mean, that sort of comes back to safety. There are often secondary exploratory endpoints of efficacy, but really more focused on the safety aspect. And in terms of redesign, what we tend to see in phase 1 more commonly is collect and hold. Sometimes if people are going to go for accelerated approval and they want some of the phase 1 patients to be part of the marketing application, then we will see that there's either a single or actually for that purpose more commonly a 2-reader with adjudication. One thing I want to point out, I think it's underutilized, that most imaging CROs can offer these services with internal staff. Just because something's collect and hold doesn't mean your images are in a vault and can't be looked at. All kinds of ad hoc analyses can be performed on these, anything from looking at a couple of patients or pulling some images to reading out the full data set. So just something to keep in mind when you're planning for early phase work. Once we get into phase 2 and 3, it's more standard as what people are used to. There's efficacy endpoints like we already talked about, like ORR, PFS, that sort of thing. And we tend to do the normal read paradigm, which is 2 reviewers read independently and come up with their own conclusions. If those differ in any key summary outcome features, like data progression, for example, then it moves to adjudication, where a 3rd radiologist looks at what both reviewers have come up with and decides which one applied the criteria more correctly. Can we go to the next slide? This just points out some of the more common criteria used in solid tumor. The workhorse is RECIST over to the left, and that's really what we see, at least as a primary outcome for most trials that are solid tumor, not CNS. But there's all kinds of modifications to RECIST, and there's more every year. The PROMISE criteria, for example, in pink is new for PSMA PET. For RECIST, there's iRECIST and IT-RECIST, but there's also modified RECIST for hepatocellular carcinoma, for mesothelioma. So we see this sometimes when people have basket trials in the phase 1 setting and they want to read out, all of these different tumor types. It can add complexity if the read criteria are not going to be the same. If we go to the next slide, we can see that we basically see the same pattern for hematologic malignancies, where our workhorse criteria are Lugano-Chesson, but then there's some modifications to those with RACIL and LYRIC. And then there's also disease-specific criteria like HALEC for CLL or IMWG for multiple myeloma. We can go to the next slide. So now I wanted to switch gears a little bit. We sort of covered the sort of general schema of how we do imaging at different states of drug development. This is a quote from the draft guidance that was released last year for the new ICH guidelines. And there's been a fair amount of discussion about this language in the imaging clinical trials community, in part because, as it says, as you read through it, it's basically saying the sponsor needs to provide the investigator with timely access to data, even if that's coming from a third party like a central lab or central imaging core lab. There is a caveat for exceptions to access in the protocol, and it says, for example, to protect blinding. So again, there's been a lot of conversation about what this means exactly, and the wording is a little bit vague. If we go to the next slide, we can see that Clario's take on this is in line with what we have been doing and what most core labs are doing, which is that there's no reason for a site to have access to all of the central review data. That really does kind of impact the blinding aspect of it. But there are things that impact patient care. There are things that, like eligibility. Is the patient going to be eligible for the trial? Does the patient have progression? Are they going to come off trial? Are they going to cross over to another arm of the trial and be re-baselined? All of these things are instances where information is fed back from the core lab to the site and the sponsor. And just to mention, I mean, there's everything from just the simple, does somebody have measurable disease by RECIST 1.1? And we're starting to see other kinds of things now as trials get more complex. So we've been asked to ascertain centrally that there are no brain metastases in a patient. We've also been asked the reverse in order to stratify patients with brain mets. As there are more and more recurrence in adjuvant trials, we're starting to get requests for central review of absence of disease at baseline. And then with some of the novel radioligand therapies, PSMA, for example, there's even more types of eligibility review where there's confirmation that there's at least one PSMA positive PET lesion, or the other thing we've seen is confirmation that no more than 50% of the visible anatomic disease is negative by PSMA PET. So these are all instances where the information would be fed back to the site from the central lab. And then confirmation of progression people are probably relatively familiar with, so you can keep going. And we can go to the next slide. I wanted to also talk about clinical data. It's another question we get a lot from sponsors, and it's something that I think some sponsors are more familiar with than others. There's a couple different kinds of clinical data that can be used to really improve the quality of the BICR output. The first is giving pre-enrollment data to the radiologists themselves, and that would include things like history of prior radiotherapy, whether a lesion that was irradiated in the past has grown subsequent to the radiation, surgical history or local regional therapy history, whether the patient has a known lesion that's been biopsy proven to be benign but could mimic tumor. These kinds of things, regulatory authorities are very comfortable with having radiologists have in advance. They don't consider that to be biasing. And it really helps with target lesion selection, and it helps align what the central reviewers are doing more with the site, where the site may not pick a very obvious lesion because they know it's been irradiated. The other kind of clinical data that's really useful, and we're seeing more and more adoption of, especially in certain disease states, like head and neck cancer, for example, is a clinical medical oncologist overread of the imaging data. So you follow the normal imaging process. It goes to 2 readers and an adjudicator if necessary. And then on study clinical data, things like a biopsy showing a new lesion as a second malignancy rather than progression, cytology data from a pluricentesis or paracentesis that shows it's acellular and not malignant. Other kinds of on-study radiation therapy, on-study surgery, these kinds of things can really help when the oncologist overreads, because they can modify the radiology response and downgrade it if it needs to be downgraded, upgrade it, or change the data progression. And what we find is that this really helps mitigate site central discordance quite a bit, especially in as I mentioned, certain diseases where the imaging can be complex, or where there are trials where there's a lot of concomitant procedures happening on trial. Next slide, please. This is just a schematic showing basically what I described. The imaging would go through the workflow on the left where the two readers read. There's a comparison of adjudication variables. If the readers both agree, the radiology assessment's complete. Reader one's data is sent to the oncologist along with all the clinical data available, and they can provide an overall integrated response. If it goes through adjudication, it's more or less the same thing. It's just whatever readers agreed upon is the one that's shared with the oncologist. Next slide, please. Now we've covered those, I wanted to just talk about some trends we're seeing in the imaging space. Obviously, everything's always going faster and faster, especially since COVID. So we're seeing faster timelines from preclinical going into first in human, such that, I mean, it's almost worth planning a little bit of a clinical program even in sort of late preclinical phase. And then we're seeing faster cycle time just through the phase 1 through 3 cycle as well. Especially with accelerated approvals. Given that imaging is a digital endpoint, it's obviously very susceptible to advances in information technology, of which we've all seen a lot of progression in the last 5, 10, 15 years, especially with AI. And part of why artificial intelligence is important is that the algorithms make segmentation of lesions and generation of 3D volumetric ROIs versus the standard 2Ds much less cumbersome. And the amount of difficulty and time and expense required to do that type of segmentation without any type of algorithmic assistance tends to be prohibitive in the drug development area. And it ends up being hard to do some of these more advanced imaging assessments on a routine basis or in a trial setting that's not just a subpopulation. Can we go to the next slide? What we've seen in our work is that AI is helping in a couple of different ways. It's improving annotation quality, so that a lesion that's manually drawn might be a little bit different from reader to reader, and we've even seen that readers following the same lesions can come up with different outcomes of PR, or SD, or PD because of how they're drawing. And obviously, in algorithmic segmentation, while it can be edited by the radiologist, overall, we've seen an improvement in consistency and annotation quality. And as I said, because this type of automated segmentation supports volumetric endpoints, it's what allows some of the things I had listed on the last slide, things like radiomics analysis, tumor growth kinetics, total tumor burden, total metabolic tumor volume. Some of these biomarkers that are more quantitative and advanced are being made more available to us through AI-based segmentation tools. Next slide, please. I talked about radiomics already, so I think I will skip this in the interest of time. I do want to touch on this slide, though. This is a point just about the quantitation and how the AI can really add value. So in prostate cancer, there are several new response criteria, one of which is RECIP for PSMA PET scans. And there's a visual qualitative RECIP, and then there's a quantitative RECIP that involves segmentation of all lesions and classification into different areas, bone, soft tissue, lymph nodes, et cetera. And what we've found with multiple sponsors and trials is that it's on a global scale, the sites cannot do a quantitative RECIP. They just do not have the bandwidth or threshold or tools to do it. And so one method of adoption that we've seen with some of these newer criteria is that sponsors will use a visual RECIP at the sites. They'll use a visual RECIP as the primary endpoint for the central review as well, but then they'll have an ad hoc analysis as an exploratory endpoint with a single reader centrally to do the quantitative RECIP that's assisted with AI algorithm segmentation. I think I'm going to stop here. I'm going to turn things over to my colleague, Dr. Mole, who's going to discuss ePro strategies, and then we should have time for some questions at the end if there are any.
Thank you, Anu. It is my pleasure now to speak to the ECOA portion of this webinar. ECOA stands for Electronic Clinical Outcome Assessments. I would like to more specifically focus on those outcome assessments which are reported by patients, which are also called patient-reported outcomes or PROs, We're speaking about them being collected electronically, e-PROs. Next slide, please. This is giving you basically a brief historical summary about the last 20 years or so, from about 2000 to 2020. What you can see on the left-hand side is that we already can observe an increase in the usage of COAs, be it in terms of the number of oncology studies that use them, that's the bars, or the proportion of studies that are using them, that's the dotted line. So overall, oncology is actually doing quite well with respect to the adoption of COAs. This is probably related to the fact that it's a very competitive area and you want to go for the broadest benefit risk assessment that you can potentially have. It might also be related to the regulators being very vigilant with respect to capturing patient experience. However, and that's the right-hand side of the slide, and you can't read that fine print, so you have to take my word for it. It's a relatively limited number of outcome assessments that have contributed to that increase. Some of them are actually not PROs, but clinician reported. Some of them are not necessarily oncology specific, like the EQ5D. And when they are oncology specific, like the EOTC QFQ C30, they might not be tailored to the exact tumor type cancer type at stage. Next slide, please. So this is an example of something that we have been knowing for more than 50 years. There's a tendency for clinicians to rate the severity of adverse symptoms quite differently, i.e. lower, than patients would do it. This is an example in a pediatric cancer study. So every every stack presents one adverse symptom, and what you can see is that the severity ratings by the investigators, the red dots, are always lower than those which are provided either by the caregivers, that's the green ones, or by the pediatric patients themselves, the blue ones. This speaks again to the importance of capturing the patient voice in terms of patient-reported outcomes. So in summary, we're doing better quantitatively, but we need to still do better qualitatively. And this is where, on the next slide, please, a couple of recent regulatory guidances come in. These ones are either by the EMA or by the FDA. And there's no need for you to try to read through all the individual names of these guidances. Suffice it to say that they are all from the last 6 or 7 years. And if there's one guidance that you want to read through, I strongly recommend that it is the FDA guidance on core PROs in cancer drug development that has reached its final stage at the end of 2024. I think this one is so important that I'd like to spend one or two more additional slides on it. Next slide, please. And more specifically, I'd like to speak to two selected aspects of that 2024 FDA core guidance. On the left-hand side of this slide, you can see a number of core PROs, 5 to be exact, which are covering a couple of domains of interest that the FDA wants you to collect and see reported. You can find these in the guidance itself. The one which I would like to highlight, because I'm going to speak to it on a later slide, is the one in light orange, which is asking for the patient-reported capture of symptomatic adverse events. Secondly, and that's the right-hand side of this slide, the FDA insists on the need to find the appropriate frequency for collecting PROs in cancer studies. And the appropriate frequency, if you think about, for example, an infused IMP, which only requires the patients to come to the sites every 3 weeks, the appropriate frequency is not to do it only once every 3 weeks at site. So in the upper panel in the table, the FDA, for example, suggests that these adverse symptoms are being collected once every week during at least the first 2 months of your study. And that actually makes a lot of sense if you think about what you would be looking at if you only ask the patient every 3 weeks how they're feeling. So imagine that you have a recall period of 7 days. Patient comes to site and they report out before the next infusion about how they felt during the last week, which is actually the 3rd week of the previous cycle. So there's a fair chance that what you are looking at is not going to be really reflective of the patient experience throughout the entire 3-week period. And it's also not likely that you're going to see a meaningful difference with your baseline assessment. There's another slide on that a little bit later on as well. Next slide, please. This is a slide which speaks in a little bit more detail to the tool which has established itself as the standard to collect symptomatic adverse event in cancer studies. So that's the Pro-CTCAA. As its name suggests, it's also developed by the NCI. It is designed to complement the investigator-reported CTCAE. And there's also a pediatric and a caregiver module available of this assessment. How does it work? Well, you do have a library of more than 120 predefined adverse symptoms. And you can select for your study, let's say about 10, 12, maybe not more than 15 symptoms for which you're going to ask at the appropriate frequency whether your trial participant has experienced it or not. If they have experienced it, depending on the symptom, there might be follow-up questions about the severity, about the frequency, and about the impact that that symptom had on aspects of their daily life. Next slide, please. So with the Pro-CTCAE and the adverse symptoms out of the way, I also wanted to speak one more time to the aspect of the frequency of adverse event collection. This one here was a prospective study. What is interesting is actually in the bottom right panel. So what these investigators did was to compare the outcomes for an EQ5D and an ERTC QLQC30 for 2 cycles. That panel is for the EQ5D, but what you can see is that, and that's the light line in the background, if you only collect these data at every site visit, every 3 weeks, you lose a lot of granularity of the patient-reported information. On the contrast, if you give the patient the opportunity to report out every week, you see that the results are actually much more reflective of what is probably their experience while they are receiving the IMP in 3-week cycles. Next slide, please. So having spoken to this, I hope I have at least convinced you that considerations around getting the frequency right, frequency of PRO collection right, is important. You might say that, well, in this case, that's fine. We just have to give our trial participants a stack of paper with the outcome assessments on them. And that might actually be an issue because, as you can see in the left panel, if your patient has paper at their disposal in order to enter data, there's a lot of things that can go wrong with respect to usage of the assessments, with respect to when they fill them in, with respect to the clarity information, and so on and so forth. So basically handing out paper for completion outside of the trial site is taking the risk of getting data that is not really in alignment with the Alcoa principles, and regulators are aware of that. In contrast, if you implement your outcome assessments electronically, you have more control about aspects such as displaying the assessment at the right time, to the right patient, one that has been trained, and making sure that the assessment is used in the way it is intended to be. Next slide, please. The left-hand panel here is basically a wrap-up of what I just said. So we do have regulators that insist on the need to collect quality data in oncology studies about patient experience, and that highlight that electronic collection of data is the best means to ensure that the data is of high quality. In that respect, it's also important to note that the fact that electronic collection provides you with better quality data and usually faster also means that it's actually the cheaper way of collecting these data than paper would be. That has a lot of risks associated with it. Secondly, and that's the right-hand side, you can also highlight that the electronic collection of paper helps you build a study that is more patient-centric and provides direct advantages to your trial participants. The one I've already spoken to, but which I would like to highlight again, is the safety aspect. So if you collect data electronically, such as the adverse symptoms, you can implement alerts so that even if this is reported away from the site gets an alert, an email that prompts them to contact the participant. Another aspect is, again, the data quality, the cost associated with getting quality data, but also the burden for sites and for participants. So, for example, if you reduce the number of queries that you need before a database lock, it means that your site doesn't have to chase your CRO doesn't have to chase the site, and your site doesn't have to chase your participants in order to clarify data that might be ambiguous if it was collected on paper. There's one last point which isn't on that slide, and that could be a webinar in its own right. But if you have electronic data, you can also think about implementing real-time analytics on that data. Next slide, please. If you are now convinced that electronic data collection is indeed the superior methodology, that would be great. One point I still wanted to highlight, though, was that going electronic doesn't mean that you shoehorn your patients into a one-size-fits-all approach. So we have options with respect to the electronic modalities that are being used. We also have backup options in case there should be an issue with the device. And all of that allows us to make sure that the setup is appropriate with respect to the regulatory requirements, the requirements to get quality data, and the patient centricity aspects. And lastly, to wrap up, I just wanted to highlight on my last slide that we are also now starting to see PRO or ePRO based data that is being integrated into new product labels. So this slide highlights the example of a 2024 label in red positive MTC. The sponsor did collect through a one-item instrument, single-item instrument, the overall perception of treatment burden by the participants for 1 year, once per week, and found that with their IMP, they were able to demonstrate that the periods of low overall burden were longer and more extended than they were with the standard of care, and the FDA considered these data solid enough to include them into the label. And with that, I would like to hand it over to my colleague, Fabian. Thank you.
Thank you, Thomas. Let me start off by going to the next slide, discussing cardiac safety. Evaluating cardiac safety is crucial for all new drugs and biologics, including oncology medicines. Many oncology medicines have cardiac toxicities, and when health authorities review a new drug application, NDA, or biologics licensing application, or BLA, the health authorities will weigh the treatment's benefit versus the treatment's risk. For oncology drugs, the balance is shifted towards the benefit or efficacy of the drug because of the unmet medical need when treating cancer. In cases where an oncology drug or treatment is effective, the drug may have adverse effects that are tolerated and can also be managed. However, there are limits in terms of safety with regards to an oncology therapy. There is no overall benefit if your cancer improves, but the treatment causes a cardiac arrest, for example. Some oncology drugs can cause heart failure as an adverse effect. The benefit-risk ratio of such an oncology treatment may not be acceptable. The treatment may increase cancer survival a few months, but this might be at the expense of causing severe heart failure that limits the ability of the patient to walk 20 feet. Next slide. Regarding assessing cardiac safety in oncology trials, the requirement is that drug developers characterize the potential cardiac effects of a new oncologic agent in order to inform clinicians how to manage the cardiac safety of the marketed oncology drug or treatment. Prescribing information goes into the label and guides clinicians on patient selection, dosing, and monitoring of the marketed drug. And as I mentioned earlier, a tolerable and manageable degree of cardiovascular toxicity may be acceptable if the benefit risk of the drug is positive. And this is the case for tyrosine kinase inhibitors, TKIs, and immune checkpoint inhibitors, ICIs, which have been revolutionary in oncology treatment over the past few years. Many TKIs are small molecules, some are antibodies, and these inhibit VEGF or other multiple protein kinases. Tyrosine kinases are expressed in cardiac and vascular tissue, and it is possible that these drugs can have side effects, which can include hypertension, left ventricular dysfunction, QTc prolongation, arterial thrombosis, myocardial ischemia, and infarction. Immune checkpoint inhibitors are typically monoclonal antibodies that can have direct cardiac effects, and these are increasingly being used to treat a larger variety of cancers, but cardiac side effects can include myocarditis, pericarditis, Tatsukobu cardiomyopathy, which involves left ventricular dysfunction, QTc prolongation, atrial ventricular conduction disease, myocardial ischemia, and infarction. Next slide, please. How do we assess cardiac safety of oncologic agents? A mainstay is the ECG, which is common to development of all drugs. The ECG can be performed in phases 1, 2, 3, and essentially in all phases of drug development. It's useful for assessing the interval duration between the components of the ECG, such as the PR interval, QRS interval, QT interval, to assess cardiac rhythm, assess the waveform, to look for pathologies such as myocardial ischemia or infarction. A major objective of the ECG is to assess whether or not a drug prolongs the QT interval of the ECG. The QT interval being prolonged is an accepted regulatory biomarker for drug-related torsade de pointe, which is a fatal ventricular arrhythmia. Another common approach to assessing cardiac safety in oncology trials is measurement of serum biomarkers. One can measure cardiac troponin to assess for cardiac injury. One can assess BMP or brain natriuretic peptide to assess for heart failure. These are relatively inexpensive and repeatable assays from blood and can be used to detect cardiac toxicity well before imaging detects this toxicity. Blood pressure, of course, is also a mainstay for cardiac safety assessment. The goal is to detect large changes in blood pressure, and this can be performed using a variety of approaches, which I'll cover in the next few slides as well. If there's a concern regarding effect of a drug on cardiac structure or function, serial assessments using cardiac imaging are important, and this can be performed using echocardiogram, MRI, or nuclear cardiac scanning. Next slide. Assessing the QTc interval. I mentioned this in the previous slide. Shown on the left are three waveforms just to illustrate that it sometimes can be difficult to assess the QT interval because it's hard to determine where the T wave ends because of the varying morphologies of the T wave. And the assessment of the QT interval is often done using a thorough QT or TQT study, which involves healthy volunteers. However, most oncologic agents cannot be given to healthy volunteers. And so an approach that involves concentration QTc modeling strategy can be utilized in oncology phase 1 and 2 first-in-human trials. This involves triplicate ECGs matched to PK collection time points. There's an intense amount of ECG and PK assessment. It is important when doing this ECG and PK assessment that a wide range of drug exposures are covered, and especially dose exposures that will be seen in phase 2 and later, for example. It's also important to consider the effect of drug accumulation, delayed effects of the drug, such as hysteresis, and also effective drug metabolites. All these can affect the QT interval and need to be considered when executing this strategy. The goal of this strategy is not to detect a small QTc effect, as one would in a thorough QT or TQT trial, but to detect or exclude a large QTc effect greater than 20 milliseconds. And this is achievable in an oncology first-in-human study. Next slide. Illustrates some of the devices used for ECG and arrhythmia assessment. Shown on the left is a traditional 12-lead ECG, which typically measures 10 seconds. Shown next to that on the right is a pocket ECG that has an accompanying smartphone that can be used by the patient at home to assess their ECG and transmit the information to a central lab. And in many cases, a 10-second ECG is not sufficient to detect cardiac events that do not occur that frequently, and this can be achieved using a continuous ECG monitor shown in the bottom right, such as a Holter ECG, second from right, and the patch monitor, which is a very small device that can be essentially pasted onto a patient's chest wall and can be worn for 2 to 4 weeks, while the Holter is typically a 24-hour device. Next slide. Blood pressure. Blood pressure is a mainstay for assessing cardiac safety. Devices, which I'll show in a second, involve a clinic or office BP assessment, ambulatory blood pressure assessment, and also home BP assessment. The FDA has issued, the FDA recommends in general for cardiac safety assessment that this includes a consideration of preclinical and non-clinical and early clinical signals, along with the understanding of the cardiovascular risk of the target patient population. With regards to blood pressure, the FDA has issued an update in 2022 regarding their draft guidance on pre-market assessment of pressor effects of drugs. And this provides information regarding requirements for assessment of pressor effects, labeling implications of pressor effects, and also best practices on assessing pressor effects. And the FDA encourages sponsors to engage them for input on a case-by-case basis with regards to a comprehensive pressor effect assessment. However, at this point, the FDA has expressed continued interest in pressor assessments, but this is not a formal requirement for all new drugs. Next slide. Just to illustrate the devices I mentioned earlier, on the left is the office or clinic blood pressure assessment device. And the middle is an ambulatory blood pressure device that's worn by the patient, shown on the right hip of the patient with the cuff on the left. And home blood pressure device can be utilized to extend the longitudinal and serial assessment of blood pressure. These devices typically provide step-by-step instructions on the screen to help guide the patient to doing this at home by themselves. Next slide. So I mentioned earlier, if there's a concern regarding effects on structure and function, cardiac imaging serially can be used. Shown here are two apical long-axis views by echo. On the left is without contrast. On the right, you can see the contrast bubbles in the contrast in the chambers. And this allows one to assess for both structural changes, such as might be involving valvular abnormalities, and as well cardiac function. Next slide, cardiac MRI. In the next slide, provides a more comprehensive assessment of left ventricular function and cardiac structure and detailed tissue characterization. However, as you can imagine, this is more expensive and more difficult to operationalize, but can provide a great deal of additional data to address cardiac safety. Next slide. In summary, for assessing cardiac safety in an oncology program, many factors should be considered to develop a cardiac safety plan. This includes early safety signals, the mechanism of action of the drug, the target patient population of the program, and drug class effects. 5 risks to always consider regarding cardiac safety include QTc prolongation, left ventricular dysfunction, myocardial ischemia and infarction, pressure effects, conduction disorders, and arrhythmias. The assessment of QTc and left ventricular dysfunction is already a regulated area, and there, as I mentioned, there is a recent draft guidance on assessing pressure effects from the FDA. This is the completion of the cardiac safety section. I'd like to pass it back to Todd Rudo for a summary.
Okay. Well, thanks, Dr. Chen. Thanks to Drs. Benzo and Moll as well for your insights. That does bring us to the conclusion here, and I just want to highlight a few key takeaways before we get to the Q&A segment. We reviewed some of the key challenges that we face in oncology development as the why behind the innovative endpoint strategies and solutions that followed. Dr. Banzel took us through the role for centralized imaging in different phases, highlighted some of the innovative modalities that have made a real difference as well in terms of how we study drugs today. Dr. Moll reviewed why a well-defined PRO strategy is so essential and what can be done from a tech perspective to enhance the quality of the patient-reported outcome data, electronic versus paper, and the considerations for site and patient burden as well. And then from a cardiac safety perspective, you just heard from Dr. Chen on this particular challenge. There are many different ways that oncology therapies can lead to adverse cardiac consequences, from QT prolongation to cardiomyopathy, adverse effects on blood pressure. And so a tailored or targeted approach is needed to strike the right balance for your particular program. So with that summary, I appreciate everyone's time and attention. I'm glad to see that quite a few questions have come in already. Please feel free to send any additional questions in through the chat function or the LinkedIn Live portal as well. I'm going to invite the other panelists now to come back on screen with me here, and we'll go through a few questions. So this first one here looks like it's directed to Dr. Banzel. You mentioned use of early phase collect and hold imaging for exploratory analyses. Can you explain how this would work?
Yes, sure. So I did. I pointed this out earlier that it's an underutilized feature that I think most imaging CROs can provide. Most of us have internal staff, radiologists. I know we have 8 radiologists on staff who do things like this in addition to implementing BICR in trials. And so it's the kinds of requests we get, the reasons why people do this are very varied. It could be looking at just the responses from sites to confirm that they're true, that, okay, we do have 12 CRs in these specific patients. It can be pulling images and looking at specific subjects for a poster presentation or a conference. It can also be a full type of exploratory analysis. If you want a radiomics assessment of the chest lesions in all the patients, I mean, we can do something like that too. So the data's not, it's not like read it or do nothing with it. There's an in-between area where there's a lot of room for different kinds of analyses for different purposes.
Great. Okay, thanks for that. Second question here, I'm going to send to you, Thomas. You mentioned that eCOA is more cost efficient than paper, so electronic was more cost efficient than paper, but that doesn't address the problem that eCOA is complicated to implement and poses a compliance risk, especially with elderly patients. So any thoughts on that you could share?
Yes, thanks for the question, Todd. It's an interesting one. It's also interesting because actually that concern that elderly patients might not be able to use electronic devices quite often comes directly from the site personnel and is put forward as a reason why enrollment is slow. We actually don't believe that it is a true obstacle anymore. And one of the main reasons is pure mathematics. If you think about those elderly patients which we are now recruiting into clinical trials that use electronic devices, if you assume that they have, I don't know, say 65 to 70 years of age, for example, they have been exposed to portable electronic devices and touchscreens since they were 15 for the last 10 to 15 years easily. So this population is used to using these devices, and there isn't really a good reason to assume that just because of the trial context they can't do it. You could even argue probably that if an eCOA solution is designed according to best practices and a patient is not able to use it, then maybe there are cognitive deficits which would also make your patient not an ideal candidate for paper administration as well. So long story short, it shouldn't be a true obstacle nowadays anymore. Thank you.
Okay, great, thank you. Dr. Chen, this is one you touched on just briefly, and I think this individual is looking for a bit more. So can preclinical or non-clinical experiments be used to assess cardiac safety? So do we really have to rely on adverse events, in monitoring during human studies is the question.
Well, most drugs, almost all drugs, undergo preclinical, non-clinical assessment. An example would be the hERG assay, which is a non-clinical study, and they're often preclinical studies involving rodents or non-human primates. And if there are any cardiac safety signals that arise from these studies, this would certainly increase the concern for monitoring cardiac safety in clinical development. And certainly in phase 1, there are potential for safety signals that could enhance your decision to increase monitoring for cardiac safety in phase 2 and 3, for example.
Right. Okay, great, thanks. I think we'll squeeze in one more here for Dr. Banzal. This is something I think that comes up quite a bit, actually, so curious for your insights here. What happens if a site disagrees with central imaging review on eligibility or progression? So how's that discrepancy managed?
Yeah, so I mean, I can certainly speak to how it's managed in some settings. I mean, there may be different approaches at different CROs, but certainly what's commonly done is, again, using that same pool of internal staff. We will look at the imaging assessments performed by the central reviewers. We'll look at the imaging assessment performed by the site investigator as well, if that's available. Typically it is, and provides for better assessment. And then we'll compare and we'll see, listen, why did the central reviewers miss something? Should the patient have been eligible, or should they have been coded as progression? Or is this within reasonable medical judgment? Is it just because they picked different target lesions at baseline? or are they measuring a lymph node? I mean, we've seen everything. I mean, I've seen people measure the long axis of lymph nodes instead of the short axis and say somebody's eligible. So there's all kinds of reasons why the discordance happens, but usually with that sort of third party kind of look by physicians who can settle out whether the case should be reread because there's some sort of error in the BICR process, or whether there's an explanation that can be given to the site via the sponsor.
Okay, great. Yeah, that's helpful. I mean, again, a common scenario. So all great questions here. Fortunately or unfortunately, we have still about 6 or 7 that we're not going to get a chance to get to. You have our commitment that we will follow up by email to those of you who asked questions where we couldn't get to them today. Sonia, I will turn back over to you now so you can close this out. Thanks.
Well, thank you very much for answering all those questions. Just like Todd said, for those of you that did not get your questions answered, the team at Clario will follow up with you. And if you are interested in hearing more from Clario, please check out their next webinar, and you see it there, dated for Tuesday, July the 15th, Progressing Beyond Exploratory Endpoints, the Value of Digital Biomarkers in Neuroscience Clinical Studies. And you can use that QR code here or visit their website for further information. Okay, now if you have any further questions, please utilize the email address you see here on your screen, and that's science@clario.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Give me a few seconds. I'm going to share a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And let me just grab that and put it in the chat box now for everyone to see. And there you go, everyone. You should see it now. Okay, and for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as reviewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you very much, Thomas, Anu, and Fabian, and of course, my co-moderator for today's event, Todd. So thank you very much, gentlemen. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until we meet again, I'm Sonia Hunt. Bye for now. Take care, everyone. Bye.
Bye.
Bye.